Okay, now let's get this done. This is November 2023, variant 1.1. These are the latest exams, okay, published just this year. Okay. Until recently, the typical virus known to science were 20 to 150 nanometers in size. In 2003, the Mimi virus was discovered with a size of 680 nanometers. In 2013, the Pandora virus was discovered, which was a size of over 1000 nanometers. So you have here three viruses. You have the, 1 minute. Okay, so you have the typical virus, which is this size, and you have the Mimi virus, which is 680, and you have the Pandora virus, which is 1000 nanometers. Which viruses can be used using a light microscope with a maximum resolution of 0.25 micrometers and an electron microscope? Now, can someone remind me what resolution is?
It's the ability to distinguish between 2 points.
Very good. It is the ability to distinguish between 2 points. Now, if the value of the virus that we are looking at is smaller than the resolution of the microscope, will we be able to see it?
No.
No. Very good. So, first, we will notice the changes in units. We have here the virus size is in nanometer and the resolution of the microscope is in micrometers. So, first, we will do what? The units, they can be different?
No.
No,
We
they
will
have
keep
to
it.
be very good. We will have to be the same. We will always convert to the smaller unit. So what do we do?
We're gonna do 0.25 times 1000.
Very good. So 0.25 times 1000, then the value here will be 250 nanometers. Any object or any virus smaller than 250 nanometers cannot be seen. So the typical virus, it is from 20 to 150. Can it be seen?
No.
Can we see? No. So A is wrong. So I'm going to cross out A. I'm sure that A is wrong. I'll cross it out to limit the number of options. Now, the Mimi virus is 680 nanometers. Can I see that?
Yes.
Yes. And the Pandora virus is 1000 nanometers. Can I see it?
Yes.
Yes. So the answer will be I cannot see the typical virus, but I can see the Mimi virus and I can see the Pandora virus. The only answer is B. Are you all with me? Did you all understand how I got it?
Yes.
Yes.
Good.
Yes.
Okay. Now let's go to number 2. The eyepiece graticule and the stage micrometer. This is really not a nice question. The diagram shows an eyepiece graticule and a cell viewed through a microscope. 1 minute. When the eyepiece graticule was calibrated at this magnification, the whole length of the graticule shown covered 12 divisions of a stage micrometer scale. I would underline this sentence. And they were 100 divisions in 10 millimeters of the scale micrometer. Yes. And now this is the image. And the image clearly shows you that this cell is 30 divisions, 30 divisions on the eyepiece graticule. They will ask me, what is the actual length of the cell? And I have two units in the question, a micrometer and a millimeter. Okay. Now, what will you do? How will you start? Who will tell me? This question is a multi-step question. Do I help you or someone will help me? Mm, no one is giving me any answer. Okay, let's start with this sentence. There were 100 divisions in a 10 millimeter of a stage micrometer. Now, I hope you understand the difference between a stage micrometer, or you remember, and the eyepiece graticule. The eyepiece graticule, from the name, this is the ruler that is found in the eyepiece of the microscope. There's a microscope and you have an eyepiece, and there is a ruler inside the eyepiece of the microscope, and this eyepiece graticule has no units. And there is another scale known as the stage micrometer. You have this in your notes. You have like a slide with a small ruler on it. Okay, but this has a scale. Every division is a certain length on millimeters. They're telling you that there were 100 divisions in 10 millimeter of the stage micrometer. So now we are talking about the stage micrometer. Pay attention with me. There were 100 divisions in 10 millimeters of the stage micrometer. So these are 100 divisions. These 100 divisions are equivalent to 10 millimeters. So one division is equivalent to how many millimeters?
We will divide 10 by 100.
Correct. You will divide 10 by 100. So every division on the stage micrometer, don't get confused, every division on the stage micrometer is 0.1 millimeters. Good. Now, they gave you another piece of information. The whole length of the graticule, when they calibrated, when they put the stage micrometer and the eyepiece graticule and they brought them into focus, the whole length of the graticule shown covered 12 divisions of a stage micrometer. Now, the whole length of the graticule, all this graticule, is also 100 divisions. So now we're talking about the eyepiece graticule. 100 divisions on the eyepiece graticule was, or it was equivalent to 12 divisions on the stage micrometer when they were brought to focus. We said every division on the stage micrometer is how much?
0.1 millimeter.
0.1. So 12 divisions is how much millimeters?
1.2.
Yes, 12 times 0.1. So let's go back to my eyepiece graticule. 100 divisions is equal to 12 times 0.1 millimeters. We are now talking about the 100 divisions on the eyepiece graticule. So let's multiply that. 12 times 0.1, this is 1.2. So 100 divisions is 1.2 millimeters on the eyepiece graticule. We want to know the value of one division on the eyepiece graticule. So one division will be how much? When we have 100 divisions on the eyepiece graticule, it is 1.2 millimeters. So
1.2
one division,
divided by 100.
yes, 1 times 1.2 divided by 100. You are correct. So 1.2 divided by 100. The value will be 0.012. This is one division on the eyepiece graticule. Now look at the picture. This cell is how many divisions on the eyepiece graticule?
30.
30. So if one
30
division is
times
0.
0.01.
Exactly. 30 multiplied by 0.012. So 0.012 multiplied by 30. The answer will be 36. We are still in millimeters. All the units are in millimeters. To convert it to micrometers, what do we do? Come on, this is the last step. To convert it to
Divided.
micrometers. No, 1 millimeter
times
is how
قصدي
many?
times 100.
1,000 times
1000.
1,000. So the 36, 1 millimeter is 1,000 micrometers. Yes? So 36 times 1,000, the answer will be 360 micrometers. Did you understand how we got this question? Hello?
Yes.
Yes.
You all did? Sure? This question just needs to be broken down sentence by sentence. We first used this sentence to help us, and then we used this one. Do you want me to repeat it, or did you all understand it?
Miss, I understand بس 36 times 1000 هتبقى 36,000 مش 360.
Sorry, not 36. It was 0.36.
اه
Okay, 0.012 times 30. Let me do it by calculator. 0.012 times 30. Yes, it is 0.36. You're correct. Times 1,000, it will be 360 micrometers. Got it?
Yes.
Okay. There is always a question like this on every single exam. So please understand the difference between the stage micrometer and the eyepiece graticule. It's easy to remember. The eyepiece graticule is the one found in the eyepiece. It's called eyepiece in the microscope. And the stage micrometer is a scale with a measurement on a slide. Okay, you want me to show you the difference? Let me show you the difference. Look, eyepiece graticule, eyepiece graticule. I showed this to you before. The eyepiece graticule is found in the eyepiece itself. This small one is the eyepiece graticule. This small scale is in the eyepiece graticule itself. No, this is not what we want now. Look at this picture. This is the stage micrometer, and the smaller ruler is the eyepiece graticule. It's always 100 units. I want to show you the eyepiece itself in the microscope. This is the eyepiece. The normal eyepiece that you know in your microscope. Inside it, there is a ruler, an eyepiece graticule that has no units because it depends on the magnification that you choose. And the stage micrometer is on a slide like this. It's a slide. It's a slide with a ruler. It's a slide with a ruler. And this ruler, every division is 0.01 millimeters, or they give you the measurement. This is a normal slide, like a specimen slide, but there is a ruler. And we have to put this slide on the stage on the microscope and bring it into focus depending on the magnification that you chose. Okay? So please make sure that this question is understood. Where am I? What happened? Okay, we're here. Let's look at number 3. Tay-Shah's disease results in the
We
buildup
can
of
see the
lipids. Okay, oh, what happened? 1 minute. Now can you see? Now can you see my screen?
Yes.
Okay. So Tay-Shay disease results in a buildup of, this is the latest exam, by the way. This is 2023, November 2023. Okay, Tay-Shay disease results in a buildup of lipids. Which cell structure does not function correctly in this disease? Golgi body, lysosome, mitochondria, or smooth endoplasmic reticulum? Which organelle gets rid of worn out organelles in the cell? And it's not working now. This is a very easy question.
Lysosome.
Yes, 3B. I want all of you to participate, not only Noor. 4. Which animal cells would have the most extensive Golgi bodies? Ciliated epithelium, goblet cell, red blood cell, or smooth muscle? Again, this is easy.
B.
Which one produces substance that is made up of mainly protein?
B.
Yes, correct. Mucus is mainly made up of protein and water. 5. The diagram shows 3 circles, 1, 3, and 5, and the shared structures 2 and 4. Okay? Let's fine. Which row correctly identifies the 3 circles and some of the structures that are shared between them? Now, when you get a question like this, because this question is very common, always look at the 2 and the 4 and what they're sharing. So I'll go to the 2. Let's look at A. Circular DNA, is it shared between chloroplast and mitochondria? And let's look at 4. Is the 80 Vedberg ribosomes, the big-sized ribosomes, are they shared between mitochondria and prokaryotes? Is A correct?
No.
No, A is wrong. So I'm going to exclude A so I don't waste my time. Let's look at B. I'll go to the shared 2 and 4. Let's go at 2. The 2, 80 Vedberg ribosomes, is it shared between chloroplast and mitochondria?
No.
No. So I'm going to remove B, finish. Let's look at C. I'll go to the shared structure 2. The circular DNA, is it shared between prokaryotes and mitochondria? And 4, is the circular DNA shared between mitochondria and chloroplasts? Yes or no?
Yes.
Yes. Correct. Okay. So 5 is C. Okay. So 6. Which row about the genetic material in animal cells and prokaryotic cells is correct? Let's look. Animal cells, they contain linear DNA. Is this true? Animal cells, do they contain linear DNA? Come on. Yes or no? In our cells, is the DNA linear or circular?
Linear.
Linear, of course. So D is wrong. I will remove D. It's easy to exclude out letters. Prokaryotic genetic material is surrounded by a double membrane. Come on, is this right or wrong? In bacteria, is the DNA surrounded by any membrane anyway?
No.
No.
No. Okay. No. So I see that C is correct. Prokaryotic genetic material is double-stranded DNA. Yes, they have circular DNA and it is double-stranded. It is made, any DNA is made up of two polynucleotide strands anyway. The only difference is that in prokaryotes and bacteria, they are not surrounded by a nuclear membrane. So 6 is C. Are you all with me? Am I moving too fast?
No.
Is everything clear? Good. 7. What is present in all viruses? Uracil, ribose, thymine, or guanine? Viruses, in case you forgot, are either DNA or RNA. So what is present in all viruses? Is uracil present in all viruses?
No.
No, because if the
Guanine.
virus is a DNA, it's not. Yes, you are right, guanine. Ribose is only found in the RNA virus. So if we have a DNA virus, ribose will not be present. So B is wrong. Thymine is only present in a DNA virus. So it is not right. So it's guanine. 8. The test for non-reducing sugars requires a second Benedict test to be carried out. Focus. We are here doing a test for non-reducing. What is the test for the non-reducing sugar? We heat it with what? We boil it with what?
Acid.
Very good. We boil it with hydrochloric acid. This will provide the water needed for the hydrolysis reaction so that the disaccharide is broken into two monosaccharides. And then we will cool it down and we'll add any alkaline. So it is neutralized. And then we will add what? The Benedict and carry out the Benedict test as normal. So which set of steps and the correct method for carrying out the non-reducing sugar test before carrying out the Benedict's test? Perform the Benedict test, which will give a negative result. Warm gently with dilute hydrochloric acid and then neutralize with sodium hydrogen carbonate. A can be right or wrong.
Wrong.
Okay. B, you said right or wrong, Mariam? I did not hear you.
Wrong.
Wrong. There is something wrong here, a little bit wrong. What was it?
Warm gently. It's supposed to
Yes.
boil.
Yes, we don't warm gently. So I will exclude A. B, perform the Benedict test with the color of the solution remaining red. Boil with hydrochloric acid and then neutralize with sodium hydrogen carbonate. B is right or wrong?
Wrong.
Wrong, because the Benedict test will give me a negative result. It will not be red. Perform the Benedict test with the color of the solution blue, remaining blue. Boil with hydrochloric acid and then neutralize with sodium hydroxide, sodium hydrogen carbonate. Is this right or wrong?
Yes, correct.
Yes, correct. Are you all with me? Let's look at D. D has to be wrong. Perform the Benedict test, which will give me a negative result. Neutralize with sodium and then warm gently. Of course, the order here is incorrect, so D is wrong. Are you all with me? Okay,
Yes.
9. Now, if I'm moving too fast, tell me to slow down. The diagrams show 3 types of covalent bonds. We have this one and this one and this one. Now, let's look at 3. 3 is the easiest to identify. We have a carbon bonded to a nitrogen. This is which type of bond? Where is it found? Come on, come on. When the carbon, the nitrogen is found where?
Proteins.
Yes, so this is which type of bond? What's the name of this bond?
Peptide.
Yes, peptide. Wake up. Okay. Now here, when we have the oxygen bonded to a carboxylic acid, this carboxylic acid group, the C double bond O, is found where? Is it found in carbohydrates? You forgot. The structure of the amino acid. Let's remember together. This is important. The structure of the amino acid. We have a carbon bonded to NH2, an amine group, and then to another carbon. This central carbon is bonded to a hydrogen in a variable R group, and the other carbon here is another functional group. It is bonded to a carboxylic acid group, the C double bond O, OH. This is called a carboxyl or a carboxylic acid group. And this carboxylic acid group is found in another molecule that we studied. Who remembers? The fatty acid. Come on, wake up. The fatty acid is a chain of hydrocarbons that can be saturated or unsaturated with two functional groups. At one end, you have the CH3, the methyl group, and the other end, you also have a carboxylic acid group. So the C double bond O here is found in either fatty acids or amino acids. Okay? We don't have the C double bond O in the sugars. Do you remember the sugar? Remember the glucose? Let's say I'll draw the alpha glucose. This is my ring. 1, 2, 3, 4, 5, 6. Oxygen, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. Carbon 6 is outside the ring, CH2OH. This is not a carboxylic acid group. This is a hydroxymethyl group, CH2OH. No C double bond O. This is not present in sugars at all. Carbon 2, we always said we'll start with carbon 2. OH down, OH up, carbon 3, OH down. And then we'll go back to carbon 1 because it's variable. Either the hydrogen is above in alpha glucose and the OH is below, or the opposite in beta glucose. So I'm reminding you with the basic molecular structures that you have to know. Let's look at the question. Which bonds will be found in glycolipids and glycoproteins? Glycolipids. 1 minute. Be patient, please, because I don't like the past papers here. I don't even have it. Okay. Which bonds will be found in glycolipids and the glycoproteins? Let's look at the glycoproteins. In other words, a protein bonded to a carbohydrate. This will be which bond? The one that has a protein. Come on. Bond 1 or 2 or 3?
Protein 3.
Yes. Okay, thank you so much. So protein will, so I will look for glycoprotein. So the answer is A or B. Okay, I'm going to exclude C or D. Glycoproteins only. Now bond 1, the easy bond, the one with a C and a double bond oxygen. This is found in either proteins or fats. So it will be A or B, glycolipids only, or glycolipids and glycoproteins.
B.
Okay, but let's go to bond 2. Bond 2, this is carbon, oxygen, carbon. This is a sugar. This sugar can bond with a lipid or a protein. This sugar itself can bond with a lipid or a protein. So 2 can be found in glycolipids and glycoproteins, or one of them, or both of them. This sugar can bond, if I choose B, B says that bond 2 is in glycolipids only. This sugar only bonds to lipids, it does not bond to proteins.
It bonds to protein.
It bonds to proteins. Why bond 1 is glycolipids only? Because there is no nitrogen in bond 1. So it is not a protein. Are you with me? So the answer 9 is A. Let me get a better exam that is not messed up like this. Okay, I'll open this exam from another. Yes. Okay. Now, this was question 9. Oh my God. This was question 9. Let me erase. It doesn't erase. Okay, who has problems with this question? Because this question is not nice. Look at this question. The diagram shows 3 types of covalent bonds. And you have to be very, very familiar with the bonds in order to answer this question. This question, if you go to bond 3, you see that there's a nitrogen. So this will immediately take you to a peptide bond, a bond that is found only in proteins. So bond 3 has to be glycoproteins only. So the answer here will take us to A or B. That's why I will exclude C or D. Okay? Now let's look at bond 1. This C with a double bond to the O is found in fatty acids and found in amino acids. But here there is no nitrogen. So this is a lipid. So that is why A will take us to glycolipids only. So this is going to narrow us down to option A. Let's go to bond 2. This bond, this is a sugar. Carbon bonded to oxygen and then to another carbon here. This is a sugar. So this sugar itself can bond with a protein or bond with a lipid. That's why bond 2 can be glycolipid or glycoprotein. And the answer for 9 is A. Did you understand how we got 9? Does anyone have any questions? Is 9 confusing? Can you give me an answer? Look.
I understand. We understand.
Okay, I want to show you something because I went back and looked at the notes. There was here, where am I? Yes. There was actually a table that I realized has exactly a summary of all the bonds. This is cell structure. This is not what I want. Okay, where is it? Here. There, you have it in your notes, this page that shows you a summary of all the covalent bonds in organic. Please pay attention to this table. Carbohydrate C, carbon bonded to oxygen, bonded to carbon. There is no carboxylic acid here. The carboxylic acid, the C bonded, double bonded to oxygen, is in the proteins and in the fatty acids, in the lipids. Okay, so this table is very important. This is exactly what you had in this question. The nitrogen, immediately when you see a nitrogen, it's a peptide bond. When you see a C double bond oxygen, and this C is bonded to an oxygen, there is no nitrogen here. So this is in a lipid, ester bond. C bond oxygen, C, so the oxygen is in the center. This is in a carbohydrate, a glycosidic bond. I hope this clears because this question is very tricky. Okay? Let's not waste time. So we already reached 10. That's very good. Which molecules would be found in an oil, not in a fat? Now, what's the difference between the 2 types of fats here? You have liquid fat and solid fat. Remind me, I forgot.
Liquid fat is unsaturated fatty acid, and the
Correct.
solid fat is the saturated fatty acid.
Good. So the liquid fat, this is the unsaturated. So what do I expect to see in the structure, on the hydrocarbon chain?
Double bonds.
Double bond. So I'm going to look at every structure. This is the hydrocarbon chain here, and this is bonded to a glycerol, fatty acids and glycerol. You should be familiar with this now. And 2, I see a double bond here. 3, I don't see a double bond. 4, I see double bonds. So the answer will be 2 and 4. The answer for 10 will be D. Clear? Are you all with me? Okay. 11. Which row about the bonding found in the primary, secondary, tertiary, and the quaternary structure of the protein is correct? So, the primary, actually, covalent is right or wrong? and peptide, I have two options, either covalent or peptide. They're both right or they're both wrong, or what? What would you do? Wake up with me. I'm not going to give you all the answers. In a primary bond, in a primary structure in a protein, what is the only bond that is found?
Peptide.
Peptide. But isn't it also a covalent bond? So the peptide bond is covalent or it is an ionic bond?
Covalent.
Of course, covalent. All these organic molecules, the bonds are covalent. So the primary here can be either covalent or peptide. So in my opinion, all these options are possible if I look at the primary. Now the secondary bond, which is the alpha helix and the beta pleated sheet. What is the main bond that will hold it in place?
Hydrogen.
Hydrogen. So this is going to narrow down my results to either B or D. I'm going to exclude C and exclude A. Okay, now the tertiary. I have all the bonds. Yes? Hydrogen, ionic, covalent, and hydrophobic. Quaternary, all the bonds. Hydrogen, ionic, covalent, and hydrophobic. Here, look at, this is B. Look at D. D, they tell you in the tertiary and the quaternary, I have ionic and disulfide only, or the quaternary, I have ionic and disulfide only. Is D right or wrong?
Wrong.
Wrong. So I will now have only one option, which is B. Okay? If you want to make sure, let's look at the mark scheme. Where is 11? Is this the correct exam? No, this is June 2019. No, sorry. Okay, this we are doing November. Which exam are we solving? November 23, 1-1. So mark scheme 1-1. We said 11B. 11B, yes. Okay, let's put this here. Okay, we did 11 questions. That's very good. By the way, the exam, the first 20 are easy. The last 20 are difficult. The specific heat capacity of different substances is shown. What is the specific heat capacity? Remind me. It's the amount of temperature needed to do what? We took it in biological molecules, also in biological molecules, in water.
We change by 1 degrees Celsius.
Very good. Okay. Which statement is correct? Air is a more stable environment than water because it is more resistant to changes in temperature. Look at air and look at water. The specific heat capacity is 1 and 4.2. Is A right or wrong? I don't know why it's taking you so long. Is A right or wrong?
Wrong.
This, of course it's wrong. It's less stable. B, it takes more energy to raise the temperature of hydrogen gas than it does to raise the temperature of water. Let's go to hydrogen gas. Specific heat capacity for 14.3, water only 2.1. B, right or
Correct.
wrong? Correct. Okay, let's not waste time. Finished. 12B. You want to make sure? 12B. Yes, we are doing a good job. Okay, 13. Graphs, important. The solid line on the graph represents the product formed over time for a reaction in a cell. Which other line represents the effect of adding the enzyme for this reaction? So the solid line, this is the solid line. Where is it? This is the solid line here. It represents the product formed over time. When we add an enzyme, how will this affect the total product form? Will it be C or D or A or B? Now, when we add an enzyme, what does the enzyme do? Make the reaction faster or slower?
Faster.
Faster. But the total product form, will it give you more product or the
No,
same
less.
amount of product? Exactly. It will give you the same amount of product, but the product will be produced faster. So it will be a faster initial rate of reaction. Is C correct? Look at C. C will give you a slower initial rate of reaction and then the same product. Is C right?
No.
No. I want everyone to participate. Now let's look at D. Compare D with this one, with this one. D, you have a faster rate of reaction, yes, but the total product produced is less. Is D correct?
No.
No. Now A, I have a faster rate of reaction and then the same amount of product used. Is A right or wrong?
Right.
Yes, A is correct. Now let me see, who do I have here? I have Noor and Yassine and Adham and Maryam and Kareem. So why is only Noor the one who's doing the talking? 14. The graph shows how the rate of an enzyme-controlled reaction is affected by the change in temperature. All other variables are standardized. You have temperature on the x-axis and the rate of the reaction and region X. What is the factor limiting the rate? Adham, what is limiting at region X? This is easy. This is O level. Adham?
Yes, miss.
Okay, what is limiting the rate of the reaction at region X?
Temperature.
Correct. It's the temperature. If anyone has any questions, stop me. How will the removal of a reversible, non-competitive enzyme inhibitor affect an enzyme catalyzed reaction? Now we need to remember together. The non-competitive enzyme, it binds with the active site or an alternative site.
Alternative site.
Correct. What does it do to the initial reaction rate and the Michaelis constant and the Vmax? Remember the table? What does it do to the initial reaction rate? The non-competitive inhibitor.
Slows
Both
it down.
inhibitors, actually. Yes, both slow it down. So lower initial reaction rate. And what does it do to the Michaelis constant, to the affinity of the enzyme?
It
The
decreases.
non-competitive. No, it doesn't. It is only the
الافينيتي
competitive. No, the
طيب
non-competitive enzyme here does not affect the affinity or the Michaelis constant, because once the non-competitive inhibitor really is released from the alternative site, this is the active site, and this is the alternative site. If this inhibitor, if it goes out, the enzyme will immediately go back to its substrate. So the affinity doesn't change. The love of the enzyme to the substrate doesn't change. There are just circumstances, remember? But when we have a competitive inhibitor, this is my enzyme, and this is my substrate, and this is the competitive inhibitor, the enzyme can bond with the competitive inhibitor and leave the substrate. So the affinity of the enzyme to the competitive inhibitor is the same as the affinity of the enzyme to the substrate, which will lower the affinity of the enzyme to the substrate. It's like we gave this example. You have one man, which is the enzyme, and he has two pretty women, and he's choosing between them. Okay? So he loves both of them. So this means that his love to the substrate will decrease because there is another competitive inhibitor that is also as pretty. So the affinity of the enzyme will decrease in the case of a competitive inhibitor. But in the case of a non-competitive inhibitor, the affinity or the love of the enzyme to the substrate will not be affected. There are just some circumstances that are preventing the enzyme from binding with the substrate. Once these circumstances leave, the enzyme will go to the substrate straight away without thinking. Are you with me, Noor? Do you remember this?
Yes, I
Okay.
remember.
So the non-competitive inhibitor, its affinity or the Michaelis constant will not be affected. But what will happen to the Vmax? In the presence of a non-competitive inhibitor, it will decrease. Let's remember together the graph. Let's go to enzymes because I'm revising lesson by lesson. Here, this graph is very important. Where is it? The graph for the inhibitors. Here, you will have it here in your notes. You have here, this is the normal graph, the green one. The competitive inhibitor and the non-competitive inhibitor, both of them, they have a slower rate or initial rate of reaction. They both decrease the initial rate of the reaction. The competitive inhibitor, as the substrate concentration increases, it will cancel out the effect of the competitive inhibitor, and eventually we will reach Vmax. But in the non-competitive inhibitor, if I increase the substrate concentration, it will not cancel out the effect of the non-competitive inhibitor. So the Vmax is decreased, but the Michaelis constant does not change. This table here is very important. That table here that compares between the competitive and the non-competitive inhibitor. In the case of a competitive inhibitor, both of them will lower, it will lower the initial rate of the reaction, same Vmax, and reduced affinity when I have competitive inhibitor. Non-competitive inhibitor, the affinity is not affected. Same affinity, same Michaelis constant. Okay, but the Vmax will decrease. Now this answers this question. Here, how will the removal of a reversal non-competitive enzyme inhibitor affect an enzyme catalyzed reaction? Non-competitive. Non-competitive. Michaelis constant doesn't change, but the Vmax is reduced. Choose A. The Michaelis constant will decrease and the Vmax will increase. Is A right or wrong?
Wrong.
Wrong. B. The Michaelis constant will increase and the Vmax will not change. B right or wrong?
Wrong.
Wrong. C. The Michaelis constant will not change and the Vmax will increase. Right or wrong?
Right.
The Michaelis constant will not change and the Vmax will increase. Okay. How will the removal? Yes, when we remove it, it will increase. Yes, correct. So 15C, are you all with me? Did you all understand this question? This question is important. When we solve
Yes.
something, please highlight it. Okay. Highlight the difficult question. 16. The dimensions of three agar cylinders, X, Y, and Z, are summarized in the table. What is the correct order of the surface area to volume ratio for the cylinders from smallest to largest? Smallest to largest. Pay attention. Now, unfortunately, this question tells me that you need to know the surface area of a cylinder. You need to know the surface area of a cylinder. But there is a very easy way to know the smallest surface area. Smallest surface area, smallest to largest, not the largest to the smallest. So this means if we are looking for at an object with a small surface area, this object will be big or small?
Big.
Big. Okay. So let's look at X, Y, and Z. And look at the radius. Which one has the biggest radius? Z. So Z will have the smallest surface area or the largest surface area?
Smallest surface area.
Yes. And then what will follow Z?
X.
X. And then Y. This is one very easy way to know the smallest to the largest surface area. And there's another way you can calculate the surface area. The surface area of a cylinder is 2 pi r squared plus 2 pi r h. We took it in cell membranes. And I actually remember telling you, they will usually give you the surface area in the question. I told you, don't remember the surface area of the cylinder, but I am taking this back. No, remember the surface area of the cylinder because you might get a question like this. And in that case, they will not give you the formula. Okay. So there are two ways to get the answer to this question. Either I'm going to look at the radius and I'm going to look at the largest radius. The largest radius means it's a big cylinder. So it will have the smallest surface area. So Z will be the smallest surface area, followed by X, followed by Y. Or I can calculate it. So the answer will be 16C. Do you all know how we got it? 16C. Do you all know how we got it, or who's confused?
No, I understand.
Okay, the rest. Maryam, Kenzie, Adham, Yasin. Did you all get it? Can I get an answer?
Yes.
Okay. We're nearly halfway. 3 individual plant leaf cells were each placed in a different solution for 30 minutes. Each solution had a different water potential. Which row correctly shows the change in volume of the 3 cells after 30 minutes? This is easy. Now, when the plant leaf cell is placed in a water potential that is lower than the cells. In order to understand, make a small drawing. This is the plant leaf cell, and it is put in a solution that is lower than the cells. The water potential here is lower. So here the water potential is high. So what will happen to the change in volume of the 3 cells after 30 minutes? Let's look at A.
Decrease.
It will decrease. And then water potential of solution, will it be equal to the if it's equal to the cells, it will increase? No change. Yes. So A is wrong and C is wrong. The answer is B or D, because when it is equal to the cell, when the water potential in and is equal to outside, there will be no change. Now let's see, is the answer B or D, or who will tell me?
B.
Let's look at B. Yes, it's very easy. If the water potential outside is lower and inside it's higher, then what will happen? Water will move from in to out and it will decrease. So the answer is B. Are you all with me? 17B. Adham, Maryam, Kenzie. I know we are all fasting. Are you all with me?
Yes.
Yes.
Okay, you're all sleeping. Which row is a representation of one chromosome at the beginning of the prophase of mitosis and the number of DNA strands? This is very easy. How many DNA strands here? In prophase, they still did not split. Kenzie. Kenzie? Yasin? Maryam, can you tell me what's the answer for 18?
A.
No. Why A? How many strands do we have? We have 1, 2, okay. And each one of them is 2. Like this is one molecule, and every molecule has 2 strands. Isn't it every DNA strand is made up of 2 strands that are anti-parallel? This is one chromosome and another chromosome. And every chromosome is made up of 2 DNA strands. So I have 1, 2, 3, 4. 1, 2 here and 2 here. Don't you know the structure of the DNA? Let's look at the mark scheme. If we argue, let's look at it's 18, 18, 18B. 18B. You all got it? The number of DNA strand. We have 1 strand and we have an anti-parallel strand in every DNA molecule. So I have 2 here and I have 2 here. Did you get this, Maryam?
Yes.
Okay. Now, which events are part of mitosis? Interphase or telophase or cytokinesis? This is so easy. Kenzie?
C.
No, cytokinesis is not part of mitosis, Noor.
It's 2 only.
Yeah. It's only 2, yes. Okay. 20. We are midway. The statements are about genes and proteins involved in breast cancer. The protein coded by the BARC1 gene inhibits the growth of the breast cancer. Inhibits. And the protein coded by P53 gene suppresses tumors. Which combination of genes is most likely to result in breast cancer? A, when both of them are mutated. B, when one is mutated and one is normal. C, when one is normal and one is mutated. D, when both of them are normal. Which one will cause cancer? This is so easy.
C.
When both are mutated?
Yes, of course, when both are mutated. When both are healthy, it will not cause cancer. The photomicrograph shows a cell during mitosis. Now, again, this is very easy. What is happening in the cell? Chromosomes are condensing, or they are moving to opposite poles, or spindle microtubules are shortening.
C.
Yes. I hope it's clear for everyone. That's very easy. Here, chromosomes are not condensing. They condense in prophase. Okay, which diagram correctly shows the structure of a piece of DNA? Again, this is easy. Let me narrow this down. Is A correct? Look at the bases.
No.
No,
Has
A,
uracil.
we don't have uracil in DNA anyway. B, is B correct? Look at the DNA strands. Are they antiparallel?
No.
No. So B is wrong. Look at C. They are antiparallel, yes, but are the bases correct? Is this
No.
complementary base pairing? Also no. A doesn't bind with C. D, they are antiparallel, and the bases, are they correct?
Yes.
Yes. So it's D. This is easy. Okay, I think we got this question in one of the mock exams. Did you receive, I corrected 4 papers. Did you receive your grades on Google Classroom? I corrected Yasin and Kareem and Noor. Let me see. And Abdullah. I'm going to correct the rest today. So you should all have received a message on Google Classroom. A detailed message. How much did you get in paper 1 and how much did you get in paper 2? Okay, 23. Bacteria are grown in a medium containing the heavy nitrogen. After several generations, all the bacterial DNA contained the heavy. Some of these bacteria were transferred to a medium containing the common isotope of the light nitrogen. The bacteria were allowed to divide once. The DNA of some of these bacteria were extracted and analyzed. This DNA was all hybrid DNA containing equal amounts of 14 and 15 nitrogen. The remaining bacteria were left in the medium with 14 nitrogen and allowed to divide one more time. The DNA of some of these bacteria were extracted and analyzed. What is the percentage of the hybrid DNA? So we need to understand the question. At the beginning, all the DNA was heavy, N15, N15. And then they replicated. Once they were put in a medium containing the light, we did this before. So they replicated one time and then one time. When they are surrounded by the light, then you will have 115 will make 114, and then 115 will bond with 114. Are you with me? So they're all hybrid. 100% hybrid. Are you with me?
Yes.
Okay. And then
Yes.
we will make them replicate again. Again, they are surrounded by only the light nitrogen. So this 15 will bond with the 14. This 14 will bond with the 14. This 15 will bond with the 14. And this 14 will bond with the 14. So I have 2 that are hybrid and 2 that are light. So what is the percentage of the hybrid? 2 and 2. A, B, C, or D? Mariam. Do you understand
B.
the question? Yes, it's B.
Doctor, can you do the question again?
Of course I can. Okay. They said, Yasin, that we had or we started with heavy DNA that contains heavy nitrogen. And then we will put it in solution that contains the light nitrogen, the typical experiment. Okay? So I have 15 and 15. DNA nitrogen, 15 and 15. I'll surround it with light nitrogen and it will replicate one time and then the other time. When it's replicated one time, this heavy nitrogen will bond with the light nitrogen. Are you with me? Because that's the only nitrogen
Yes.
available.
Yes.
Yes? So all the DNA will be hybrid. 100% of the DNA will be hybrid. Now they will allow it to divide one more time. What will happen? This heavy nitrogen will bond with the light one. And this light one will bond with the light one. And this heavy one will do semi-conservative replication and fuse with a light nitrogen, and this light one will fuse with a light nitrogen. And they want you to know the percentage of the hybrid, not the mix. Let's look at the hybrid. This is one hybrid, this is one hybrid, and the other two are 100% light. So I have 50% hybrid DNA and 50% light DNA. Do you see that?
Yes, I understand.
Okay, very good. Now 24. Which statements correctly describe the process of transcription? Messenger RNA is decoded by a ribosome to produce a specific amino acid chain, or a section of DNA is converted to RNA by RNA polymerase, or transfer RNA transfers amino acids to the ribosome. Which one is the correct answer? This is easy. Transcription only. Mariam, where's Kenzie? She's online, but I did not hear her voice. Adham, what is the answer?
Yes, miss.
Where were you? What's the answer to number 24?
Answer to number 24. Just a second. B.
B?
لا 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 not B, not B. Um, yeah, it's not C or D.
Then what is it?
هي B, هي B, هي B.
No. We're talking about transcription. Making the messenger RNA, it's two only. A section of the DNA is converted to RNA by RNA polymerase. A1 is translation. When we
Okay.
produce the amino acid chain, this is the translation step.
تمام.
Okay, Adam?
Okay.
You forgot. Transcription, I will make a copy on the messenger RNA. And the translation, this copy will go to the ribosome and will be translated into an amino acid sequence. You need to study. Okay, 25. Sickle cell anemia is caused by a mutation in an allele of the gene that codes for the beta globin. The diagram shows the sequence of bases in a small section of the template strand of DNA for both the normal and the sickle cell globin alleles. So this is the healthy and this is the sickle. Now we took this before. We took that the GAG, the GAG, will be converted to GTG in a substitution mutation. A will be replaced, base A will be replaced by base thiamine. Both the polypeptides for the healthy and the sickle hemoglobin are the same length. How will the mutation in the allele result in the production of an altered version of the beta globin polypeptide? Now, let's look at each one of them and we have to see which one of them is correct. A transfer RNA molecule with the anticodon GUG will form hydrogen bonds with the altered codon on the messenger RNA. So this means that we have to get the messenger RNA and the transfer RNA for the sickle cell hemoglobin. This is the mutated GTG on the DNA. What is the messenger RNA for that? What is the codon? The complementary codon, the messenger RNA for GTG. If you're not going to participate with me, I'm going to leave. Because I'm not speaking to myself here. Adham.
Miss, I'm lagging بس معلش.
Okay, tell me, what is the messenger RNA? GTG is the DNA. What is the messenger RNA for that?
Messenger RNA.
Yes.
CAC.
C,
A.
A,
C.
and C. Okay. What is the transfer RNA for that?
Transfer RNA. GUG.
Yes, G, U, G. Is A correct now? A transfer RNA molecule with the anticodon GUG will form hydrogen bonds with the altered codon on the messenger RNA. Is A right or wrong?
Correct.
Yes,
Correct.
correct. Yes, it is correct. Finished. It's easy. Oof. Now let me choose someone. Who do I have? Yassine. Yassine, are you
Yes.
there? Okay. 26. The plant diagram of a transverse section through a dicot plant stem was drawn by a student. They had stained the section with a chemical that stains lignin. Which row is correct for tissues W and Z? That's easy. W. W is the phloem or the xylem?
W is the phloem, Z is the xylem, and the stained tissue is the xylem.
Correct. So the answer is
B.
B. Correct. 27. Now this is for who? Let me see. Mariam. Kareem, are you there?
Yes, miss.
Okay, Kareem, can you tell me number 27? Which route must water travel through in a plant using the symplastic pathway? Do you remember, Kareem, the symplastic pathway? Who
Yes,
remembers?
miss. Yes.
Remind me, Habibi. Tell me, what is the symplastic pathway?
Is when the water moves through the apoplast or symplast or
Yes, what is the symplast?
ال-
I'll give you two options. When it passes through cell walls and intercellular spaces, or when it passes through the cytoplasm and plasmodesmata.
intercellular spaces.
No, my dear. It's the apoplast, where the water will move from cell wall to cell wall and intercellular spaces. And
ماشي انا ما شفتش انه هو ماشي خلاص.
okay. So, number one, when the water moves from the soil into the root hair cell vacuole, this is apoplast or symplast? Mariam, can you help him?
is simple apoplast.
Habibi, it will enter the vacuole, so it will cross a cell membrane. It will enter a cytoplasm. So how can one be apoplast? You're giving me a hard time. Okay, who will help Kareem? Mariam, are you
Me.
there? Okay,
Yes.
help him. Can you please answer? One is apoplast or symplast?
Symplast.
Symplast, yes. Two, from the soil into the root hair cell walls.
Apoplast.
Yes, so we don't have to. Three, from the root hair cell walls to the root cortex cell walls.
Apoplast.
This is obvious. Okay, so no three. Four, from cortex cell walls into the endodermis cells, into the cell.
Symplast.
Apoplast, symplast. Okay. Five, from the endodermis cell into the xylem vessel.
Symplast.
It should be symplast because it will cross the cell. From the leaf mesophyll cell walls into air spaces.
Apoplast.
Yes.
لا ولا ايه؟
Yes,
مش
yes,
الاير
yes.
سبيسز.
Cell walls, air spaces, intercellular spaces, apoplast.
اوكي.
Okay. So we want the symplast. So this, in my opinion, will be 1 and 4 and 5.
مفيش one
Oh,
for
this is what's the answer?
I think 1 and 4.
Yeah, I would choose also 1 and 4. Let me see. No, let's go to the mark scheme. Oh, it's discounted.
Oh.
This rarely happens. So 27, obviously there was a lot of argument over 27, so it has been discounted. This is not common. So 27 is not going to be included anyway. A plant leaf seen in a transverse section with a microscope shows the features. A thick waxy cuticle, sunken stomata, rolled leaf. Now this is easy. Mariam, what is this leaf adapted for? to decrease carbon dioxide or increase carbon dioxide or decrease water loss by transpiration or increase water loss by, that's easy. Mariam.
Yes, increase water loss by transpiration.
Mariam.
Sorry, decrease.
Yes. Okay. Kenzie, are you there? Where's Kenzie? Do I have Kenzie here with us? No. Yassine. What occurs as carbohydrate is taken out of a sink into a phloem sieve tube element? The water potential in the phloem will become higher or lower when you have the phloem and the sugar is being loaded. So the water potential will increase or decrease?
To decrease.
It will decrease, correct. And what will happen to the volume of the liquid in the sieve tube element when it decreases? So this means it's either C or D. What will follow?
The volume of liquid. Doesn't water from the xylem move into the phloem?
Correct. So the
So
volume
the
of the
volume of
liquid
liquid
will...
increases.
Excellent. Very good. So the answer is D. Well done. Okay, Adham. Which factors, we have the last 10 questions, and then I'll leave you alone. Which factors affect the blood pressure, Adham? Diameter, systolic pressure of the heart ventricles, and the volume of blood returning to the heart in each cardiac cycle.
A.
Yes. You know why I would choose A? Because 1 and 2 are definitely correct, but I don't find an option with 1 and 2 only, so it's A. Okay. Noor. Sulfame is a molecule that inhibits carbonic anhydrase. Which effect would sulfame have inside the red blood cell in muscle tissue that is respiring at a high rate? Can you remind me, Noor, what is the function of the carbonic anhydrase anyway? What does the carbonic anhydrase do?
Catalyze the reaction between carbon dioxide and water to form carbonic acid.
Correct. Okay. Now, this, which effect will sulfame? Sulfame, it will inhibit. I don't have this enzyme. So I don't have the carbonic acid, CO3. And this carbonic acid dissociates into the hydrogen ions and the hydrogen carbonate ions. So this reaction will actually slow down. So let's see. A, it will decrease in the rate of the formation of the hemoglobinic acid. Is this correct?
Wrong.
Why wrong? Why? We have here the carbonic acid that will dissociate into hydrogen ions and hydrogen carbonate ions. These hydrogen ions, what is their fate? Don't they react with hemoglobin? The hemoglobin acts as the buffer to prevent a decrease in the pH of blood. And when they do, they form the hemoglobinic acid. Doesn't this happen? Yes
Yes.
or no? Okay. When there is no enzyme, will this happen?
No.
No. So A is right. It will decrease the rate of the formation of the hemoglobinic acid. Yes? Look at the rest. A decrease in the pH. This is not going to happen. So there will be no decrease in pH. This reaction will stop. So B is wrong. An increase in the rate of the chloride shift. No, it will not increase it. It will decrease it because this reaction also will stop. An increase in the dissociation of oxyhemoglobin. On the contrary, we don't have this reaction. The hemoglobin will dissociate with the oxygen to take the hydrogen ions. No hydrogen ions are being made because the reaction is stopped. So the only answer is A. Did you get it?
Yes, miss.
Okay, good. Let's finish the other 9 questions because, okay, my blood pressure is starting to rise. The partial pressure of oxygen in blood vessels in different parts of the body affects the percentage saturation of each hemoglobin molecule. So we have different blood vessels. We have an active muscle and a resting muscle and an arteriole in the lungs, and the partial pressure of oxygen, it is increasing, obviously. And the saturation also is increasing as the partial pressure is increasing. Now, this is the oxygen dissociation curve, if you remember. Okay? Here we have the partial pressure of oxygen, and here we have the percentage saturation of hemoglobin, and the curve looks like this. Which statement explains the difference in saturation between a resting and an active muscle? So the resting muscle and the active muscle, it increases. The saturation of hemoglobin is increasing. So we're reading the curve in this direction. We are in this part of the graph. Who will remind me, how do we read this part of the graph? We always used to start with a small change. Huh? Remind me. Maryam. I'll cry. Maryam,
Yes, doctor.
this part of the graph, how did we describe it? We explained it so many times. Slow, saturated, steep. What did we say about this part of the graph?
It's
Yes,
steep.
we said a small change, a small increase in the partial pressure of oxygen results in a great what?
Saturation of hemoglobin.
Yes. Now let's look at one of the options that matches what we say. A small decrease in the partial pressure changes two atoms of oxygen to leave each hemoglobin molecule. Is A correct?
No.
No, A is not correct.
No.
Now let's see B. A small decrease in the partial pressure between 4 to 7 causes two molecules of oxygen to leave each hemoglobin molecule because the oxygen dissociation curve is steep.
Yes.
Yes. Okay. B is very similar to what we said. C, loading of oxygen is slower between 4 and 7. Is this true? Is the oxygen loading slower?
No.
No, actually, it's more. So C is wrong. Loading of oxygen is faster between 4 and 7 due to the binding of one only oxygen molecule. Is this right or wrong?
Wrong.
Wrong. So the answer is B. Let's just make sure because we are jumping. We said 32B. Yes, 32B. Very good. Which statements about the Bohr shift are correct? Adham. The shift is accelerated by the action of carbonic anhydrase. A decrease in blood pH will cause oxyhemoglobin to dissociate. A decrease in carbon dioxide concentration will cause more oxygen to bind to hemoglobin. Let's look at each one of
B.
them. Adham, are you with me?
Yes, yes.
Yassine, are you there?
Yes,
Can you
yes,
hear
I'm
me?
here.
Doctor,
Yes, I can see your screen. Yeah, miss.
Are you with me? Hello?
Yes.
Can we finish this exam, please? Which statements about the Bohr shift are correct? Maryam, tell me. The shift is accelerated by the action of carbonic anhydrase. A decrease in blood pH will cause oxyhemoglobin to dissociate. A decrease in carbon dioxide concentration will cause more oxygen to bind to hemoglobin. Is any one of them wrong?
Doctor, you're not sharing the screen.
Is it shared now?
Yes.
Yes. Is one of them incorrect, or they're both, or the 3 of them are correct? Is one right or wrong?
It's right.
Yes, one is right. 2, a decrease in blood pH will cause oxyhemoglobin to dissociate. Is this right or wrong?
Yes, it's right.
Yes, it's right. 3, a decrease in carbon dioxide concentration will cause more oxygen to bind to hemoglobin.
Correct.
Correct. So the answer 33A. Okay, it's A. 5 more, 5 more. Let's make it. Some of the events during a cardiac cycle are listed. Yassine, what is the correct
Yes,
sequence of events after the sinoatrial node sends out a wave of electrical activity? After the sinoatrial node, what is happening, my dear? The left
the
and the
left
right
and right
atrium.
atria contract,
Correct. So we'll start with A or B. Let's exclude C and D. Huh? Quick.
and then a wave of electricity is like passes to the atrioventricular node.
Correct. So 1 and 4. So the answer is
B.
B. Okay, stay with me, Yassine. Oxygen and carbon dioxide enter and leave the blood in the capillaries of the alveoli. Which statement about this process is correct? Oxygen and carbon dioxide only pass through one cell layer. Is this right?
No.
No. So A is wrong. Oxygen moves out of the blood and carbon dioxide moves into the blood during
No.
gas exchange. No, of course not. Oxygen rapidly diffuses into the alveoli and carbon dioxide out of the alveoli due to the large surface area. Is this right or wrong?
No.
Huh?
Uh, it's wrong because oxygen doesn't diffuse into the alveoli.
Okay, let's look at D. The capillaries have the lowest blood pressure in the body, which allows the blood. Is the capillaries having the lowest blood pressure in the body? It's the veins. In my opinion, it's C. Let's look at the mark scheme. Oh, 35 is also discounted.
This discounted. Yeah.
Nice. 36. Which statement is
Yes,
correct?
why are they discounted?
Someone has a very strange. Discounted means that it's not included. It's not going to be graded. So there's something wrong with the question itself, not with you. 36. Cholera is caused by the virus Vibrio cholera. Is this right?
No.
No. B. Mosquitoes are pathogens that cause malaria?
No.
No, it's the Plasmodium. Mycobacterium bovis only cause TB in humans?
No.
No.
No. Plasmodium vivax is an example of a proctist?
Yes.
Yes, the falciparum and the vivax and the malari. 37. The graph shows changes in the antibiotic resistance of a species of bacterium between 2000 and 2016 in one country. Samples of bacteria are collected every year from 48 hospitals. The bacteria were tested to see if they showed resistance to 5 different antibiotics. My God, you have to have the graph in front of you. What can be concluded from the data? Overuse of the ciprofloxacin has caused antibiotic resistance to increase by more than 20% between 2000 and 2016. I have to go to the graph and look at ciprofloxacin. Ciprofloxacin is this graph, and we will look at its increase. Actually, we don't have numbers. Like, can you know if this increased more than 20%? Is A right or wrong? Can someone give me an answer? B, okay, you read and you tell me A, B, C, or D, because I'm exhausted. I'll read it with you. B, percentage resistance to 3 of the antibiotics was at peak at 215. I'll go to 215. This is 1, this is 2, this is 3. B looks correct to you or not?
Yes.
It looks correct. Changes in treatment guidelines have caused resistance to some antibiotics to decrease from 215 to 216. 215 to 216. Okay. There is a change, there is a decrease, but why do I know that this is due to change in treatment guidelines? Maybe it's due to better hygiene or isolation. So B is the most correct. Let's look at B. This is number 30. What? 37B. Okay, نغش 37A. يلا كويس. What's 38? Some responses made by cells of the immune system to a pathogen are listed. Noor. 3 more questions. Which responses are for the B lymphocyte? This is easy. The B lymphocyte, when a pathogen enters the body, what happens to it?
A.
Yes, مشيناه. But it's easy. Clonal selection, clonal expansion, blah, blah, blah. Okay, Mariam. Which statements about the use of monoclonal antibodies are correct? Do they give active immunity?
No.
No. Can they be injected to treat viral infections?
Yes.
Yes, why not? Okay. Can they be used as a diagnostic test for specific pathogens?
Yes.
Yes. So it's a C, easy. The last question. Which statement explains how a vaccination program can control the spread of an infectious disease? Unvaccinated individuals are less likely to meet a vaccinated individual? It doesn't look right to me. What do you think?
Wrong.
Unvaccinated individuals are less likely to meet an infected individual. Vaccinated individuals cannot catch the disease. Is C right or wrong?
Wrong.
Why not? No, why wrong? I'm vaccinated, but I can catch the disease, but I will not show signs and symptoms. Vaccination does not prevent me from catching the disease. It just gives me immunity. You understand? So C is wrong. D, vaccinated individuals, they have natural active immunity?
Wrong.
Wrong. So I have to choose between A and B. A and B, to me, they're both not very nice. But what would be the best one? A or B? Everyone will be vaccinated. So is A correct? Unvaccinated individuals are less likely to meet a vaccinated individual? A is right or not? When all the population is vaccinated.
B. B.
B, it's B. Okay. This exam was not difficult, by the way. In my opinion, the questions that were tough was this question, the question about biological molecules with this question. So mark it, the graph. Any graph question is important. And the surface area question, you have to know the formula, okay, or use the easy method that I showed you. So question, please write it down. Question 13, I'll write it down somewhere. So I will tell you the numbers. Question 13, question 15 was not easy. And the second question, so the question 10, and question 9 was not nice. Question also 2. This question, the IP's gratitude. Okay, now I'm sure that you're not depending on me. I'm sure that you are solving on your own. Like, this is what I'm hoping for. You need to start solving paper 2 more than paper 1 because paper 2 is coming first. But paper 1, you do more mistakes than paper 2. And you have 40 questions. I want you, while you're solving exams, to try to aim for 37, 30 out of 40, minimum 30 out of 40.
Miss I have a
30 out of
question.
40. اتفضل.
انا كنور اذاكر
اتفضل.
يعني اذاكر ولا احل بس بيبر دلوقتي؟
لا تذاكر انت. Your exam was very poor.
اه ماشي
It showed from your answers that you did not even know the information.
حاضر.
Okay? Do both, but you need to study. Okay, I'm going to correct the remaining mock exams and I'll send you the grades today. Meet you Wednesday. On Wednesday, we'll solve paper 2, but of course, from here until Wednesday, you solve. Don't wait for me.
Okay.
Bye-bye.
Bye.